name is Jeet and I'm going to talk about the random amplified polymorphic DNA technology. Now, for this technology, no detailed knowledge of DNA sequence is required because what we need basically is just 10 more primer which we are going to select from a DNA which has been amplified by a PCR. We are going to select the primer randomly and we are going to use it for this technology. This technique fundamentally scans the genome for these small inverted repeats and amplifies intervening DNA segments of variable length. The amplification of profile depends upon template primer combination. Now, here I would like to add a few more things about the RAPD technique, and which is about developing the local specific codominant markers by the RAPD, in which we amplify the DNA strand by PCR and then we make them longer and more specific so that it increases the specificity regarding the template DNA. Now, the applications of the RAPD. The first one among the two sets of the applications which I am going to talk is phenotypic and molecular identification of lactobacilli which is secreted from the vaginal secretion. In a particular study which was carried out by Diminito et al., the lactobacilli species were secreted from the vigorous samples and characterized. A broad spectrum of activity and nutritive properties of these vaginal tissues have been reported. Various genotypic and phenotypic methods are applied for lactobacilli species identification. These are the two plates which show, the first plate shows the vaginal microflora of the lactobacilli and the second one is just a lactobacillus species. Now, the methods involved in this are, firstly, we characterize lactobacilli phenotypically, then we analyze them by PCR, and then we carry cluster analysis and 16S rDNA sequencing and the amplification, and then we carry out the RAPD pattern by using the M13V primer. Now, the second set of application is generation of specific marker to discriminate bacillus anthracis from other bacteria, for example Bacillus cereus, because there is not much differentiation in between these two bacillus. In order to establish a method to specifically detect the beta anthracis in situations such as environmental contamination, use of RAPD PCR has been proven very effective. Now here there is a RAPD pattern, the bands are developed, and this first shows the base pattern of the Bacillus anthracis and the second one shows of Bacillus cereus. Results include the RAPD PCR fingerprinting, which is done by using the RAPD PCR method with a random 10-mer primer. The RAPD PCR band shows that Bacillus anthracis-like members of Bacillus cereus produce fingerprints very similar to those of conventional Bacillus anthracis strains. These isolates may represent links between members of group and common ancestor.
As my colleagues have told you about the traditional methods, I will be focusing on modern sequencing technologies, out of which my main focus is Sanger method and 454 sequencing. Now, this is Sanger method. It was first detected in 1977, and from till that time, the basic methodology of Sanger method has remained conceptually unchanged. This method depends on DNA polymerase dependent synthesis of a complementary DNA strand in the presence of 2 deoxynucleotide DNTPs and 2 3-dideoxynucleotide DDNTPs. The reaction synthesis is randomly terminated when there is addition of DDNTP to the growing oligonucleotide chain, and the shortened products, truncated products are obtained with appropriate DDNTP at their 3' terminus. The separation is achieved with the help of polyacrylamide gel electrophoresis, and the terminal DDNTPs are used to reveal the DNA sequence of the template strand. Now, this is what the Sanger method is. What the thing which has put this Sanger method in the modern sequence technology is that in the earlier times there were 4 DDNTPs were required to stop the oligonucleotide chain reaction, but advancement in the fluorescent techniques have made only one reaction with the help of fluorescent labeled dyes. And the second thing is the slab which they have used in the polyacrylamide gel electrophoresis has changed to the capillary gel electrophoresis for giving maximum peak of current to mark the separation matrix. Now, Now moving on the second technique, that is 4-5-4 sequencing. Now the 4-5-4 sequencing is divided into two parts, that is sample preparation and pyrosequencing. Sample preparation involves following steps like attachment of DNA fragments to the beads. These adapter ligated DNA are attached to the DNA beads, and then amplification of the DNA is done, which remains attached with the DNA beads, and then these beads, adapter ligated DNA beads, are then loaded into the picotiter plate along with the help of following enzymes such as polymerase, ATP sulfurylase and luciferase. And this whole procedure and this whole master mix is then kept in the FLX instrument, out of which when we add the nucleotides like A, T, G, C, so the nucleotide when the complementary nucleotide will come in contact with the template strand, so the polymerase will extend the DNA and give the light signal. This light signal will be detected by the CCD digital camera in the FLX instrument, which makes this technique as an example of pyrosequencing. Now, the applications of this technique. First main application of this technique is in the field of epigenetics, which is the most widely field which is related to human epigenome project. That is DNA methylation profiling by bisulfite DNA sequencing. There are various methods to detect the DNA methylation, out of which one is by bisulfite DNA sequencing, in which they have used the Sanger method, due to which the study generators have given the more than 1600 sequences from one clone rather than traditional methods which was obtained only 20 sequences from one clone. The other applications are for mapping histone modifications and the location of DNA binding proteins. And the third one is to study DNA accessibility and chromatin structure. For studying DNA accessibility, nucleosomes are the main factor which can be easily detected